Isometric and isotonic contractions are the two common types of muscle contractions. The one that you probably think of most often is an isotonic contraction. This is when the muscle tension is going to be equal or greater than the resistance, and there's actually a movement that occurs. So we see an example here of somebody doing a lifting a barbell of elbow flexion of a 10-pound barbell. There are two types of isotonic contractions. There is one that's called an isotonic concentric contraction, and there's also one that's called an isotonic eccentric contraction. The other type of muscle contraction is what's called an isometric contraction. And during an isometric contraction, this is when there's actually no movement that occurs. And this could be for a couple different reasons. It could be that you are trying to move a force that is too great to actually move, or you are deciding to hold your arm in an isotonic movement. So tension is generated, but there's actually no movement that actually occurs in this example. So you should be aware of these two examples. So in isometric, the length of the sarcomere actually does not change, but during an isotonic contraction, the length of the sarcomere does change. So let's look at the skeletal muscle fiber organization. And there's several different characteristics that you should know. And we name muscles based on the pattern of the fascicle arrangement, one of which is circular, and this could refer to sphincters. One example would be the muscle that is often used to close the eye, called the orbicularis oculi. Another example would be if all of the fascicles run parallel to the long axis of the muscle, and the muscle has what's called a central belly. A convergent type of organization is where there's a common attachment at the tendon where there is a collagen band. Because remember, a tendon is the point of attachment of a muscle to a bone. There's another type called the pennate, where the fascicles tend to resemble a feather. And in this case, the most common of the pennate is actually called the bipennate, where the muscle fibers are on both sides of the tendon, and it's the most common. There's also a unipennate and a multipennate that is also present. Our next slide is kind of summarizing and showing you some examples of these. So the circular one would be found in sphincters, and there are sphincters in passageways throughout the digestive system, for example. Also examples to orbits like the mouth and the anus. And one example would be a muscle called the orbicularis oris. The parallel, an example of this would be the biceps brachii muscle, and also the rectus abdominis, where we can see the fascicles running in a straight line parallel to the long axis of the muscle. Convergent is where there's a triangular muscle with a common attachment site. And an example of that would be the pectoralis major. Another example would be the pennate, and those include examples like the rectus femoris, which is bipennate, the deltoid, a multipennate, and the extensor digitorum, which is unipennate. Remember, these are ones that resemble kind of a feather. Actions of skeletal muscles are kind of defined with these various characteristics. For example, the muscle responsible for producing the movement is also called the prime mover or the agonist. And in this case, the triceps brachii is the agonist for forearm extension. The muscle that is opposing these actions would be called the antagonist. So if the triceps were the agonist, The antagonist, which is usually on the opposite side of the bone, would be the antagonist for forearm extension. A synergist is a muscle that assists the agonist and includes stabilizing fixator muscles. When we name muscles, we name them by different criteria, such as the action. For example, the pronator teres helps to pronate the forearm. We name them for specific body regions, like the tibialis anterior is on the anterior portion of the tibia. The pollicis muscle would be named for its attachment to the thumb. We name them for attachments, like the sternocleidomastoid is named because it attaches to the sternum, clavicle, and mastoid process of the temporal bone. So a lot of times when we break down the muscle into pieces and parts, we could kind of figure out what the function of that muscle is. Another way that we name muscles is their muscle fibers. Like the rectus abdominis, we saw that the fascicles ran in a straight line. According to the muscle shape and size, includes the trapezius muscle, which is shaped like a trapezoid, the adductor magnus, a large leg muscle. It's called magnus because it is large. Another example would be the amount of proximal muscle heads or proximal tendons. So the biceps brachii has 2 heads and proximal tendons, whereas the triceps brachii, as you could probably guess, has 3 heads or tendons or origins. The last type of muscles discussed in this chapter, we've talked mostly about skeletal muscle, but the other examples are cardiac muscle cells. You've seen micrographs of them, and you should be able to identify them. Cardiac muscles are striated, have 1 or 2 nuclei, and they form Y-shaped branches. So they're the only ones that are branched. They have intercalated discs, which are not present in the other 2 types. And those discs are made up of what's called a gap junction and a desmosome. It's not important that you remember gap junction and desmosome. And the other thing is they are autorhythmic. They automatically start to contract on their own because they are controlled involuntarily by the autonomic nervous system, which you'll learn about later on in the semester. So this is showing you a nice micrograph in the upper right-hand corner of cardiac muscle. You can see the intercalated discs shown here. They go in the same direction as the striations, but there is not nearly as many of them. In this diagram, it highlights the intercalated discs as well as down here. Notice it has the same structures of the sarcomeres, like the A bands, the I band, and the Z disc. But the smooth muscle does not have a Z disc, and that's an important thing to remember. It still has the thick and the thin filaments, so the myosin and the actin, but no Z disc. Smooth muscle, as you can see, has no striations in the micrograph. It has 1 centrally located nucleus, and it's found in the walls of the viscera and blood vessels.